This lecture will go through the first half of chapter 9, which covers lifespan development. So the chapter is entitled Developing Through the Lifespan. Um, this is a branch in psychology that talks about how we develop over time. How does our development differ as we age? So developmental psychology, you might hear it as lifespan development, is a branch in psychology that really looks at, as humans, how we change physically, cognitively, and socially through the lifespan. You do not need to know this for the test, but it looks at, let's say, cognitive development. How does our thinking and problem solving change from 2 to 4 to 6, on and on. If you had taken this course, I would say 20 years ago, we would have started with when the baby's born. We would look at their cognitive behavior, physical development, all the way up to death. However, we know whatever happens prenatally in utero, it will have an effect on the child's consequent development. So we're going to start off with conception. The three general areas that we're interested in studying is nature versus nurture, continuity versus stages, and stability and change. The one you need to know for the exam is the first one. The other two are super interesting, but for our purposes, we're going to stick to the first. Right here, I have a YouTube, which I will put up for you all on Canvas to look at. It's going to be hard to hear through this mode. But nature and nurture really looks at our genetic and environmental factors. So please make sure what you know what each one means. Nature refers to your biological genetic predisposition. Biological genetic predisposition. Nurture looks at environmental factors. So what, let's say someone like Jeffrey Dahmer, who we ultimately we know as a serial killer. What made him become a serial killer? Was it already in his nature? Was it in his biology? Was it in his genetic makeup? Or did the environment or nurture made him who he was? There's also another field that is really looking at how do genes interact with the environment to influence development. So someone like Jeffrey Dahmer, if you look at it from what we call epigenetics, was it already in his gene and something in that environment turned the gene on? For the longest time, psychology thought of these two as separate. Now we're beginning to see the interaction between nature and nurture in shaping who you are in terms of intelligence, height, everything you can think of. So what you will see on the YouTube link that I put up is a study that was done with identical twins, and how these identical twins were different or the same as they grew. In a long time ago, when the dinosaurs roamed the earth, they used to separate identical twins, meaning if there were two twins and two other families wanted to separate them, they would give one identical twin to one family and another to another family. Well, this created the perfect environment to study nature and nurture. We know identical twins in terms of nature, genetic makeup, they're exactly the same. So with the same makeup, they were put into different environments. So the interest here is how much are they alike, which will speak to nature, and how are they different, which speaks to nurture. So you will meet the gem twins, which are they're just odd, but it really highlights the nature-nurture dilemma. So please look for that. I'm not going to ask you about continuity and stability question. Basically, they're asking, is our growth gradual? Do we gradually grow, or are they in abrupt stages? And stability and change basically speaks to, are we the same as we grow, or do we change as we age? So with conception, you have a single sperm that penetrates the outer coating of this egg. Once it releases its genetic makeup, hopefully you saw that in the video, once it releases its genetic makeup into and combines, fuses with the eggs, you have one cell which is 46 chromosome. So what we understand about human genetics was from this gentleman, an Austrian monk by the name of Gregor Mendel, who did his really famous pea experiment. Let me go back for a second and just kind of expand on this. Before Gregor Mendel came, the idea was that we're all, our inheritance, our characteristics are all from divine intervention. So if you have brown hair, brown eyes, that was God's will for you to have those characteristics. Gregor Mendel came and showed that our characteristics are in fact inherited from the egg and the sperm by his pea experiment. Those of you who will be taking genetics, you might hear about Punnett square. That's Gregor Mendel, or Mendelian genetics. This is the guy who really taught us about inheriting certain characteristics. The field of epigenetics look at your geno and phenotype. For the exam, you do not need to know epigenetics, but you do need to know what geno and phenotypes are. Genotype is your genetic makeup. So if, let's say, someone has brown eyes like myself, but my father has green eyes, I may have the gene for that recessive gene for light color eyes. So even though I can't see it in my genotype, that recessive gene is present. So genotype is really your genetic makeup, what you really can't see, but it's in your genes. Phenotype, however, is the one that's expressed. So even though I might have a recessive gene for green eyes, my eyes are brown, thus my phenotype, the individual's express trait, is my phenotype. Now, epigenetics is a really interesting subfield that has come out that looks at the same genotype and how that genotype can lead to different phenotypes. And by that we mean, let's say, I'm sure you all heard of Angelina Jolie, who had the genotype for certain types of cancer. I think it was, I can't remember exactly, which was like 60% chance of getting those ovarian, uterine, and breast cancer. But it was, let's say, 60%. There's still 40% chance that she wouldn't have gotten it. So what in the environment contributes to that 60% so that she would ultimately develop those cancer? And that's what epigenetics looks at. And now it's looking therapeutically on how we can manipulate your genes to either suppress certain gene expressions or even change them so your genes do different things. It's a fascinating field. If anyone is interested in genetics, you might want to get into that. So just a quick overview of genetics. Please make sure you know Gregor Mendel, the Austrian monk, the picture that I showed you. He is considered the father of genetics because of his pea plant experiment. And what he basically showed, he combined, actually they were green and yellow for our experiment. We'll make them these pea plants that had either brown seeds or white seeds. So he took a brown seed and a white seed and combined them together, and he saw that all the offsprings, all the baby peas, were all brown in the first generation. Then he took that generation and crossbred the two browns, and what he realized is 3 of them were brown, 1 was white. So this pea experiment basically showed us that we have the idea of dominant and recessive genes are coming from the pair of chromosomes that we have. So we know the egg that we get from maternal side has 23 chromosomes. So the egg contains 23 chromosomes. The sperm contains 23 chromosomes. So they have outlined the chromosomes from 1 to 23. Please make sure you know this for the exam. From the first to the 22nd chromosome, we call these autosomes. These are chromosomes that are identical, exactly the same. So if you have, let's say, a disorder like Huntington disease, which is found on the fourth chromosome, it is also referred to as autosomal, autosome, autosomal disorder. You don't need to know that. But 1 to 22nd autosomes, the 23rd chromosome is super special because it will determine the sex of the baby. Thus is called sex chromosome. Um, Now, what's interesting, I think is interesting, is the egg, our floating egg, always carries an X on its 23rd chromosome. The sperm, however, depending on which sperm you get, one sperm will carry an X and another one will carry a Y. Either or, hopefully, in normal situation, that's what should happen. So one sperm will carry an egg X, another one will carry a Y on 23rd chromosome, depending on which one penetrates the egg. So if you have a sperm that carries an X, you have an XX, which will give you a female. An XY will give you a male. And that's all determined by the 23rd chromosome. So let's say conception has happened, the egg and the sperm come together, create the first cell. Now the prenatal stage has started. The field has divided up prenatal development in 3 stages. Most of you know prenatal development in trimesters, first, second, third trimester. We know it as stages in lifespan. So zygotic or germinal is the first stage. For the exam, please make sure you know the name, which is in blue, the time span, and then we'll talk about what is important in each one. So just a quick review. Zygote or germinal stage, zygotic or germinal stage, is from the time of conception, the minute the egg and the sperm meet, until about 2 weeks. And during this time, the egg is flowing, I will show you that in a second, and multiplying and differentiating. From about the 2nd week to the end of the 8th week, you have embryonic stage. At this point, the group of cells or the embryo is attached to the uterine wall and is beginning to share nutrients with the parent. And the last stage is from 9 weeks to birth. All the internal organs are beginning to further develop and hopefully become functional by the time the baby is born. Now, we say till birth. A full-term pregnancy, and write that somewhere. A full-term pregnancy is 40 weeks. So gestation of 40 weeks is considered a full-term pregnancy. Although anything after 37 weeks is considered full-term, for our class, when they determine when the baby's born or the anticipated birth date is going to be 40 weeks. So let's begin with the first stage, which is the germinal stage. It's from the time of conception, the minute the egg and the sperm meet, until implantation, which is about the 2nd week. Now, implantation is when this group of cells implant, burrow, or attach themselves into the uterine wall. So for the exam, please make sure you know what the word zygote is, because we're going to play with this word over and over again. Zygote is simply fertilized egg. When the egg and the sperm come together, create the first cell, that's a zygote. This zygote ultimately will divide repeatedly, become specialized, differentiate into different organs and cells, and ultimately implant itself into the uterine wall. You don't have this picture, I'll show you a picture in a second. So here we have an ovary. Every 28 days, an egg is fertilized and will release into the fallopian tube. As it travels, if it is attacked or fertilized by the sperm, the first cell is created, which is the zygote. And as it's traveling down the fallopian tube, it will begin to increase in cells. These cells will specialize into different functions. And anywhere between the 7th, 9th, 10th week, the cells will be strong enough to burrow themselves. I think you saw this in the video where they break out of zona, implant itself into the uterine wall. Now you have nutrients between the host and the embryo beginning to grow. Once these cells implant themselves firmly into the uterine wall, now you're entering the embryonic period, which we anticipate at about the end of the second week, beginning of the third week. So again, embryonic stage is from the time of implantation, which works out to be 2-3 weeks, to until the end of the 8th week. During this time, major organs are beginning to form. Some of the first organs that are developing is the brain, spinal cord, and the heart. These are your major organs that are developing before everything else. The head is first, brain and spinal cord, vital organs like the heart and the extremities will ultimately follow. Whether the embryo has gotten, or with the sperm, has gotten the X or the Y, the sex organs will begin to differentiate. You will not be able to see this in ultrasound, but if anyone at this point, it's already beginning to take place. Some of the things I've heard is we're all born a female. We are not born a female. We're actually dimorphic, which means we have both sex organs very immaturely ready to go. And depending on whether we get the X or the Y, either XX, the female organs will develop. So you get the ovaries, the fallopian tube, uterus, and the rest. And if you get the Y, it will cause the testes to develop, increase in testosterone, and all the other sex organs will develop as a result. Not that you can see it here. If the Y doesn't do what it's supposed to do, which you saw in the video, the gene segment SRY that ultimately creates the testes and onward, then the default is a female. Please make sure you know these 3 for the exam. The embryo is suspended in amniotic sac filled with amniotic fluid. This amniotic fluid is really important in protecting the organism from bumping and hurting itself, for lack of a better word. So it's kind of floating up in that amniotic fluid, and the amniotic fluid will keep the temperature constant for the growing organism. Very early on from that zygote, as a group of cells will turn into the placenta, super important structure that exchanges, that kind of acts as a screener between what comes into the growing organism and what is taken out. It is a highly nutrient tissue that unfortunately we throw away, but most animals will consume it after birth. The embryo is connected to the placenta through this long cord called the umbilical cord. If you've ever had the chance to see umbilical cord, it is one long vein that brings in nutrient, and you can see these 2 smaller arteries that wrap around it, which ultimately takes waste away from the organism. But the placenta and umbilical cord obviously are super important in our overall development. Umbilical cord can be anywhere from 1 feet to 3 feet long. So this sucker can be pretty long. Here we have, this is the normal pregnancy, what we should expect. The placenta is nice and up into the uterus, and you have the baby to kind of transitioning or getting ready for birth. This is the cervix, and the baby is born this way. This is a condition called placenta previa, where the placenta has formed lower in the uterus. This can be a dangerous situation for the mother and the infant because in order for the baby to be born naturally, vaginally, the placenta has to detach, come out first, and then the baby comes out, which means once it detaches, it is not providing oxygen or any kind of essential elements to the baby, and the baby is at risk of cerebral palsy and other conditions. So at the end of the 8th week, beginning of the 9th week, you're entering what we call the fetal period. This is beginning of the 3rd month of pregnancy until birth. This is really maturation and completion period. So a heart is started early on, it will finish up here. the liver started, it will finish up here. It is the longest stage, but it's basically completing, if you think of the embryonic stage as the foundation, this is building the entire house. The rest of the house is being built. Now, during pregnancy, you could have these agents, chemical, viruses, things in the environment that can ultimately impact and really devastate the prenatal atmosphere. These teratogens, if they enter the developing infant, can cause harm. So one of the most common type of teratogens is as alcohol. It really wasn't understood how bad alcohol was for fetal development until they began to observe these women who were giving birth to that were heavy drinkers during their pregnancy. These babies had some common features, and today we call these fetal alcohol syndrome. That can have abnormal physical development and more devastatingly is the mental impairment that these babies could possibly have. So some of the facial features that we see is the small eyes, the flattening of the nose. Was super interesting is this philtrum. If you all feel right between your nose and your mouth, you should have these two bumps that appear. Will babies that are exposed to alcohol, again, they say heavy alcohol consumption. They weren't really clear with what heavy is. Is that 2 glasses of wine or 2 bottles of wine? No one really knows, but they say heavy drinking can make this area smooth and extremely thin upper lip. Those are characteristics of children exposed to alcohol. Unfortunately, that's not the most devastating part. The most devastating part is alcohol's effect on brain development prenatally. These are 2 babies that passed away 6 weeks into their life. This is a 6-week-old that was not exposed to alcohol, and you can see the brain is nice and convoluted. You can see the cells are well developed. Unfortunately, this is a baby that was exposed to heavy drinking in utero. What that means is this baby will unfortunately would have never had a normal life. This brain is not able to allow the child to function what we call normally. So an area of psychology that lifespan development is fascinated by is twins, right? We know 2 twins, identical versus fraternal twins. For the exam, please make sure the difference between the 2. Identical or monozygotic. Remember we talked about zygote, right? The first cell created. Mono, meaning one zygote. From a single fertilized egg, you get 2 separate human beings that turn into identical twins. With identical twins, they should be the same sex because you only have 1 sperm that has either given an X or a Y. With fraternal twins, these are 2 separate eggs, 2 separate sperm, and this is when the sperm fertilizes the egg. It could use 2 eggs. It is basically 2 siblings happening at the same time, right? So they could be of opposite sex or they could be the same sex because the sperm ultimately will determine the sex of the baby, whether it carries an X or a Y. You could have 2 boys, 2 girls, or a boy and a girl. So the baby's born, now we're entering infancy. We know infants are born with certain reflexes. Please make sure you know what reflexes are. These are unlearned automatic responses to a sensory stimulation. So some of these reflexes will stay with us forever. These reflexes are, for example, when we smell something good, we reflexively, automatically will begin to salivate. When, let's say someone puffs something in our eye, we will blink reflexively. These reflexes stay with us. However, when a baby's born, it has a lot of reflexes. For example, sucking is a reflex that's needed for the baby in order to, for the baby to feed. If they have a strong sucking reflex, then they can breastfeed and survive. Babies that were born to heroin, unfortunately, had a really weak sucking reflex, and before we had the technology we have today, unfortunately, they passed away. So they have looked at what one of the more interesting is the rooting reflex. If you've ever touched a baby's cheek, especially when they're hungry, they frantically look for the source of stimulation. Well, that ultimately will help the baby find the nipple. So when the nipple hits the baby's cheek, they will look for the nipple in order to feed. Um, so they have looked at what area of the brain causes reflexes versus voluntary. Please make sure you know this for the exam. So we know brainstem, you should know brainstem, lower part of the brain, reptilian brain, is responsible for um, your reflexes, right? So when the baby is born, their brainstem is fully functional. So they will take a lot of responsibility. That brainstem will take a lot of our responsibility. One of them being sucking. So if you put something or a bottle in a baby's mouth, 1 month old, they will automatically suck and feed, even if they're not hungry, right? It's a reflex, it's an automatic response. Now, by 6 month old, if you give a baby a bottle, if they don't want it, they will turn their face, which means now it becomes voluntary. One of the reasons it's voluntary is that the cerebral cortex has taken that responsibility. As the baby's brain is developing, the cerebral cortex is waking up, becoming functional, thus taking responsibility away from the brainstem. So some of the stuff that as an infant was as a newborn was a reflex, walking is a reflex. If you take your newborn, let's say 1 month old, and you take pressure off of their legs, they will go on their tiptoes and will begin to mimic what appears to be stepping. But if you do that to a 6, 7 month old, if they don't want to step, they're not going to do it because now the cortex has taken over that responsibility. Please remember we are going to do a face-to-face, not face-to-face, but a Zoom conference. If any of these concepts are difficult to understand, write it down and we can revisit it. In terms of overall motor development, walking, running, sitting, they have looked at trends, and there are two general trends that a baby's physical development follows. The first one is what's called cephalocaudal trend. This is head to toe. So when you take your baby for the doctor's visit, the first thing they ask is, can your baby lift their neck, right? Should happen between 2 to 3 weeks. So the first area that develops is the neck. Then is the shoulders, the baby either turning or pushing up. Then comes complete the torso, the stomach area and up. They can turn all the way around, ultimately sitting and then walking. So this is in terms of our development, head to toe. Proximedistal means center outward. So a baby's torso, this area will develop faster than their arms. Their arms will develop faster than their hands. So this is the first area that develops. Then comes the arms, then comes the hands. It's a trend that we refer to as proximedistal, center outward. Now, what's fascinating about babies is we all have five senses, and of the five senses, they are born with four of them ready to go. They can taste really well, they can smell well, they can touch. What am I missing? And smell, hear, they can hear, smell, hear, taste, and touch. Okay, those four are ready to go. And please make sure you know this for the test. The sense of vision, their ability to see, is the weakest sense at birth. They have a really hard time seeing at birth. They're actually considered legally blind. Their distance is 20 to 600 sight. You don't need to know the numbers. Within days, infants can track moving light, so their muscles are beginning to develop. Optic nerve is beginning to develop. 3 months, they can discriminate most of your colors. What you need to know is this one. They have done testing with babies and how long would they look at a stimuli, we'll call the fixation time. And it's clear that babies prefer to look at human faces. So save your money with the mobiles. They really prefer to look at you during this time. And that'll stop as teenagers. So enjoy that. They really want to just look at your face. Babies also, you don't need to know this for the test, but they are responding to cues for depth, especially during the time they will begin to crawl. So if they see steps going down, they are likely to stop at the top of the stairs and not fall. I wouldn't take a risk, but they are able to see some depth. The next area we're going to look at is cognitive development. And this is basically looking at how kids mentally represent and think about the world. How do they problem solve? Does it change as they get older? Two individuals you want to associate with cognitive development is Jean Piaget and Lev Vygotsky. Jean Piaget was the first theorist to come up with stages of cognitive development. He really looked at kids and how their thinking and problem solving changes as they age. Lev Vygotsky was a Russian linguist who was also interested in language development, particularly, but how cognition develops. So we're going to look at these two, starting with Jean Piaget first. This gentleman right here is Jean Piaget. Jean Piaget was fascinated with kids, loved kids, would do experiments with them, fun experiments with them. And so working with these kids, he came up with certain concepts. Now, what's important about Jean Piaget, he believed that kids are, he called them natural scientists or little scientists. He believed that as long as you allow the child to explore on their own, to play, that their cognition will develop, they will become stronger and be able to problem solve. He said that we have these concepts or framework for organizing our world called schemas. Please make sure you know schemas. So let's say if someone said, hey, I've never seen a banana. What is a banana, right? What you would do is go into your framework that you have of a banana and begin to describe it. So you might say things like, banana is a fruit. That's part of your schema or understanding of what a banana is. You peel it. Monkeys eat it. Whatever your understanding of bananas are ultimately stored in your schemas. As we grow older, if you ask, let's say, a 7-year-old what a banana is, it may be a little bit more simplistic than yours. You already know it has potassium. It's good at eating, good for replenishing some of the potassium that you might be losing. So those, as we develop and grow older, our schemas become more and more sophisticated, hopefully. So how do these schemas change and get organized? Piaget said there's two things at play with schemas. You got to know assimilation and accommodation. On the test, you will see an example and you need to figure out whether the child is assimilating or accommodating. In assimilation is when the child responds to a new stimuli through existing cognitive structures. And what that means is, let's say your child has a schema for a red ball, right? You have a bottom a red ball and they always play with a red ball. Red ball is red. It's round, you bounce. So when a child sees a red round object, they'll go into their schema and assimilate that object into already existing schema, and they'll look at that red object and say, oh, it's a red ball, and this is what I can do with a red ball. So let's say you went to Ralph's and bought a tomato. When you come home, the tomato is sitting on top of your grocery and it's nice and red. It's one of those nuclear tomatoes that are nice and round. So the baby looks at it, looks at this stimuli, will first assimilate, will go into their framework of understanding and say, it's round and red. It must be a ball, right? So they might pick up the tomato and will begin to throw it on the ground because that's a red ball. So the mother might scream and say, no, that's not a that's not a tomato. That's not a red ball. That's a tomato. And the child is now confused because it's looking at it, it's red and round. It has to be a red ball. So the mother will begin to force the child to accommodate, which means creating new schemas in order to fit the new stimuli. So creating new ways to respond to objects or creating new schemas to understand this novel thing I'm looking at. So the child will accommodate, meaning now they will create a new schema for something that is red and round. Can also be a tomato, but with tomato, you can eat it. With a red ball, you can't eat it. Tomato, you can bounce it. Red ball, you can bounce it. So you begin to differentiate between these two red objects. So Piaget did experiments with kids, and he was really the first one to come up with stages of cognitive development. For the exam, you need to know the name of the stage. We're going to go through each one, but this is the name of the stage. You need to know the time span for each one. So sensory motor stage is from birth to nearly 2 years of age, and some of the new development that occurs. So as we go through, make sure you highlight it. But this is a good kind of overall view of the stages and what we can expect from each stage. So the first stage, Piaget called sensory motor stage, which spans from 0 to 2. That's really all you need to know, except one more thing I'll show you in a second. Basically, what he's saying is for the first few months of life, babies can't see. They're not very coordinated with their motor skills. They can't reach for things. They can't grab things. So the first way they explore the world is through their senses. They close their eyes, the object goes away. They taste the thing through their mouth. So the first few months of life is really exploring the world through our senses. As we begin to get more control over our motor abilities, we can sit, we can reach for things, we can walk. We begin to explore the world, combining our senses as well as our motor skills. So there's a coordination between sensory information and motor activity at this stage. Early exploration of the environment, super important. However, kids at this age don't have language. So what you'll see is if you give them an object, they'll automatically put it in their mouth and will begin to explore it through their mouth. The mouth becomes almost like the eyes. One of the things that has to happen in sensory motor stage, and please make sure you know this for the exam, is object permanence. So before 6 months of age, and this is according to Piaget, some studies are finding this is actually earlier than 6 months, but children, when they see an object, let's say you show it to them and they're fascinated by it, if you put it away, then the child will kind of lose interest. If I can't see it, it doesn't exist, right? And we're going to see object permanence play a part in the next, in our next slide. But, oh, I thought I had a picture, shoot. So object permanence is the understanding that objects remain even when out of sight. The second stage for Piaget's cognitive development theory, he called this stage, if I can get to it, pre-operational stage of development. Now, pre-operational goes from 2 to about 6 to 7 years of age. The word operational for Piaget meant logical thinking. So pre meaning before the child can think logically. So during this time, kids are depending on intuition rather than logic in order to solve problems and make sense of their world. By 2, their language is rapidly developing, so they have words and symbols to represent objects and the relationship between them. They, however, do have these 3 limitations. For the exam, please make sure you know what age span, pre-operational stages from 2 to 6 or 7, and that they're depending on intuition rather than logic to solve problems. So these 3 limitations, egocentrism, is the child's inability to take another's perspective. And you'll see this in the video. If it's not happening to them, it's not happening at all, right? So if you've ever played hide and seek with kids this age, They want to be right in the midst of things. So when they go hide, they usually go hide behind the curtain or something that you can see their legs. But if they can't see you because they're behind the curtain, you can't see them. So you find them, and then when it's your turn, you go hide in a really good place. And then ultimately, the child finds you. Let's say work in a closet, it takes them forever, but they find you. Now, when it's their turn to go hide, they will go hide the same place you did because in their mind, if it took me a long time to find this person who was hiding in the closet, it'll take them a long time to find me. So it's the inability to take another's perspective. They can only see the world from their perspective. Animism is the belief that inanimate objects have real life qualities and can move because of will or spirit. So when children, let's say they're running and hit their head in on the table, most parents will rub the area, kiss it to make it feel better. And if the child is still crying, many parents will hit the table, right? So if you look at it from a child's perspective, you table hurt me, so my mommy's going to hurt you back, right? And then they'll start hurting the table, hitting the table and saying bad table, makes them feel better. So artificialism is the last concept. This is the belief that natural occurrences are under human adult control. So your child is likely to say, Mommy, make the sun come out. I want to go out and play, right? You have control over rain, wind, whatever it is. Please change it to their liking. So some of the things, one of the ways Piaget came up with his stages was the conservation task. So what he did was he had kids, he lined up these pennies in front of the kids, and right in front of them, he expanded it, he separated them, and he said, are they the same amount of row in each, or penny in each row, right in front of the child? And kids in pre-operational age were unable to tell to see the transformation. They were unable to reverse the object back to its original size. I'm going to show you this video. Hopefully this will expand on it. So as you can see, the transformation was right in front of the kids, but because they are stuck centrated on the image that they're seeing, they're unable to transfer, let's say, this liquid back to original and know that they're the same. The next stage, Piaget called this concrete operational stage, which is from 7 to about 11. At this point, kids have logical thinking. However, and please make sure you know this for the exam, they're extremely concrete and black and white in their abilities. They can't think hypothetically, abstractly. They can do simple math problems, and they can do the conservation task. But if you ask questions like, what does it mean, metaphors, right? What does it mean, don't cry over spilled milk? They can't see beyond the milk spilling, right? So they can't understand the metaphoric meaning behind it. So here I have another YouTube. Now, what I want, if you've ever dealt with a 7, 8, 9 year old, they know everything, right? It's that awkward stage between being still a young child and an adolescent. Take a look at the kids' faces when they ask them the same questions. Brilliant kid, brilliant kid. So you can see the difference in responses between pre-operational and operational, concrete operational. And what's great about this age is you begin to see their personalities. You can really see the one that's a little bit sarcastic, all you did was smash it down, you didn't do anything, versus the one that's a little bit nicer, which was a little bit more polite. Um, the last stage, and this is where Piaget really stopped his stages, is called formal operational stage, 12 and up. He said because of brain development, because of cognitive growth, kids are able to think abstractly, imagine realities, systematically reason. This is adolescents that can read books with metaphors and um, the abstract thinking. So I'm going to show you another video and you can see the difference between the two stages pretty clearly. So the difference between the two, we go from thumb wrestling, which is concrete operational stage, to the whole world would change, much more abstract and hypothetical thinking. Um, that's really where Piaget stopped. His stages really stopped then. Lev Vygotsky is your second cognitive behaviorist. Lev Vygotsky was interested in cognitive development, but the way he thought about it was more about social interaction. So if you are, let's say, if you have a school that is more geared towards Piaget, what you will find is Piaget believed kids are like little scientists. You allow them to explore their environment by themselves, and they will develop to be great cognitive thinkers, or have a good cognitive ability. Lev Vygotsky said it's important for kids to play and explore. However, his focus, and please make sure you know this for the exam, his focus was much more on the influence of culture and children's interaction with the elders. So his emphasis was much more on talking to the kids and guiding them through problems and helping them problem solve through language and interaction. For the exam, there's two concepts you need to know. You don't need to know zone of proximal development. You can X that out. But inner speech, you need to know inner speech is simply talking to oneself, right? We all talk to ourselves. Let's say we're, especially now, scared of coronavirus and we're walking around, someone coughs, and you might tell yourself, oh, I bet they don't have coronavirus. I bet they'll be perfectly fine. So you kind of calm yourself down through that inner speech. Or when you problem solve, you might talk yourself through the problem. In scaffolding, scaffolding is assisted learning. So in scaffolding, ultimately what you want to do is help the child, assist them in solving problems, assist them as needed. So if you're giving them a puzzle, if you're using scaffolding, the first time they try to solve the puzzle, you have them, you walk them through it, right? You talk to them, this is what you do first, and then you do this, and then you do this, right? The next time you stand back and allow them to problem solve by themselves a little bit. And when they get stuck, you step in, you scaffold your assistance and help with them. He really believed a lot of the maturation that happens is due to brain. And that comes with brain maturation and kids' ability to have better insight. So we're going to stop here. This is the first half of lifespan development. Again, if you have any questions up to this point, please write it down somewhere so that when we have our Zoom conference, you have it ready to go. Hope you have a wonderful day. Please be safe, and I'll see you online.